Classical pharmacokinetics focused on and is able to simulate the absorption, distribution, metabolism, and excretion of chemical in the body. In addition, you have several parameters that can vary among populations, such as age, sex, body weight, and also exposure conditions such as the mode of administration, the mode of exposure, the level of exposure, and all of this variability could affect significantly the exposure of this population. In this context, PBPK models are able to take account of this variability, by providing physiological parameters to this model, such as blood flow, cardiac output, organ weight, organ volumes, metabolic clearance. You can have a quite accurate prediction of the fate of the chemicals in general population, but also in subpopulations, such as sensitive population, infants, toddlers, transplant patients, elderlies, pregnant women. PBPK models are actually mathematical equations that summarize what is occurring in the body, a body that is consisting in several compartments, each compartment representing an organ, and each organ is connected with a liquid, mainly blood. Well, in the context of Oberon, we have to date developed 5 models so far. We are still developing some extra models. These models, for QSAR, these models are freely available on Vega platform. Vega is a platform initially developed by the Mario Negri Institute. It is made of it provides several computational tools in order to predict the chemical properties or activities of chemicals.
So, omics studies, bioascriptomics and metabolomics, and particularly the heat regression, provide a holistic overview of the biological changes induced by the tested endocrine disruptors in the Oberon project, like phthalates, bisphenols, and PFAS. The gene expression and metabolic profiles, in fact, gave us information about potential biomarkers of exposure and actually elucidated perturbated pathways that can be served as indirect or direct key events, and that can help us make a more accurate, more precise health risk assessment, and subsequently provide more targeted regulatory strategies for protecting the human health from exposure to these endocrine disruptor chemicals that are in everyday life products. So overall, among the multiply omics experiments we performed in different in vitro models like pancreatic, liver and adipose tissue cells models, or in vivo models like 5 days and 5 weeks post-fertilization zebrafish, we found consistently perturbed pathways related to lipids and amino acids metabolism, like sphygmolipid and rigorous phospholipid metabolism and tryptophan metabolism. That needs, of course, further investigation in order to be connected with the in vitro study results.